Howdy, my future doctors. So finally, I've decided to do an educational video in this channel, and this is the second video. So it's been a while, okay, since the last time I did the first video. I wasn't being able to do another video because I'm doing my first year MBBS in the names. So I was very busy, and it's really difficult to find the time for everything. So I'm still trying to manage everything. I'm still trying to build a solid routine. It's been only a month, but still there is a pile of subjects, a pile of concepts which have to be learned. So in between all of these, but I just wanted to do consistent videos and help you guys out in all possible ways related to MBBS. Let it be the admissions, let it be which college to choose, let it be how to study or what to study, that kind of things. I'm planning to do that in my channel, this channel. So today we are going to discuss about erythropoiesis. I'm recording this voiceover separately from that class, so there might be some repetitions of these words initially, but what I'm trying to say is we are going to start with erythropoiesis. And in erythropoiesis, the first and foremost step is to get the iron for the production of hemoglobin. Sorry, hemoglobin is the most important part of RBC, right? So it is what gives RBC the ability to carry oxygen around the body. So to form this hemoglobin, the main component which is required is iron. Okay, so in this video we are going to discuss about the iron intake and how it is metabolized, how it is reaching the site of erythropoiesis. Okay, so let's get straight into it. So today we are going to try and explain the process of erythropoiesis. So what is erythropoiesis? Erythropoiesis is the process of production of erythrocytes or RBCs, red blood cells. So for this process to occur, there are some main components which has to be put together and which has to be absorbed from the diet, different types, which will eventually and ultimately will contribute to the production of erythrocytes. So between these important components, the major one, the elephant in the room, is iron. Okay, so there are two sources of iron, Fe3 plus, the ferric, and Fe2 plus, which is the ferrous. So ferric and ferrous. Normally, the iron is obtained through diet, and in diet we can obtain iron in two ways. Through diet we can obtain iron in two ways. One is heme iron and the other one is non-heme iron. Okay, so heme iron is from what you would say, it is from non-vegetarian or meat sources. And the non-heme iron is from vegetarian sources or leafy vegetables and all. So this is about the iron. So how is the iron is absorbed? That's what we are going to discuss in this video in detail. So when we consume any kind of iron, if I had some spinach and a steak today for lunch, it will have both what, the heme iron and the non-heme iron. So the heme iron is ferrous, and the non-heme ion is the heme ion is ferrous, which is Fe2 plus, and the non-heme ion is ferric, that is Fe3 plus. So I'll have both of them, Fe3 plus, which is from the non-heme ion, and Fe2 plus, which is from the heme ion. So both of these will go and through cells of duodenum. Okay, this is the part duodenum. So from here we'll take a small section of a cell, and we are going to zoom in this one particular cell here, that is this enterocyte. Okay, so that is this enterocyte. So what happens is there are two type of ions waiting here, Fe3 plus, which is from the non-heme source, and the Fe2 plus, which is from the heme source. So the Fe2 plus can freely enter into the cell because of the presence of this green transporter, which is known as DMT1. Okay, then what do you mean by DMT1? DMT1 stands for divalent metal transporter 1. So DMT1 is divalent metal transporter 1. So through this divalent metal transporter 1, the ferrous form of ion enters the enterocyte. And now what happens to this ferric form? It will go to another protein on the membrane, which will actually make this ferric ion to a ferrous ion. So what process is happening here? It is a reduction process. So this protein or this enzyme is a reductase. So this enzyme is cytochrome reductase B. So cytochrome reductase B, or duodenyl cytochrome reductase B. Okay, so what happened to the ferric form? It is also converted to the ferrous form. Now it can be easily transported inside the cell through this DMT1 transporter. So inside the cell there will be this proteins which are known as apoferritin. Okay, these apoferritins will bind to these newly coming Fe2 plus ions, because this Fe2 plus ions cannot be let to roam around the cell or in the body anywhere, because it will cause free radical reactions or free radical production, which is very detrimental for the body function. So this ion will attach to the apoferritin and will form what you call ferritin. Okay, so and these ferritins will cluster together. All of these are ferritins with, like, apoferritin with iron Fe2 plus is ferritin, and the ferritins cluster together to form, this cluster is called hemosiderin. Okay, now from the ferritin, some Fe2 plus will have to go out to the bloodstream. So where is the site of production of RBC? It is in the bone marrow. So what we need our raw materials to reach the bone marrow. How can we reach the bone marrow? We can use the blood supply to the bone marrow to transport all the essential components or the raw materials needed for the erythropoiesis to the bone marrow. So we'll have to get this Fe2 plus, or essentially the iron, to the bone marrow through blood. So to let it into the blood, the iron must move out of this cell, which is the enterocyte of the duodenum. So the Fe2 plus will move out through what? Again, a transport protein, which is known as ferroportin. Okay, so it is a protein called ferroportin. Through ferroportin, the Fe2 plus will go out, but still there's the problem of Fe2 plus starting some free radical reactions. So to avoid that, we'll use this protein here to convert it into ferric form. Okay, and this protein is known as hephaestin. Okay, but the process of the ferroportin is controlled by another protein, you may call it. Okay, so which is that protein? That protein is called hepcidin. So this hepcidin can actually inhibit the ferroportin. So you might ask, why should we inhibit the ferroportin? If the ferroportin is overly active and it just letting the iron to move out freely, what happens? The amount of iron in the blood, Fe3 plus, will increase. Okay, and that is not good. That condition is known as hemochromatosis, overload of iron in the blood. So to avoid this condition and to keep the ferroportin in check, the hepcidin helps. Okay, and this hepcidin production is regulated. The hepcidin is produced in the liver, and the production of hepcidin is regulated and controlled by a protein called HFE protein. This is that protein, HFE protein. Okay, HFE protein. So this HFE protein is homeostatic iron regulator protein. So we'll write that here, HFE protein, which is it's not hemostatic, it's homeostatic iron regulator protein. So HFE protein is homeostatic. This is homeostatic iron regulator protein, and this protein is coded by a gene, the HFE gene, which is present in the short arm of chromosome 6. So any mutation of this one gene will cause the production of a defective or no production of this HFE protein. So the absence of HFE protein will affect the production of hepcidin, which in turn will affect the function of ferroportin, and which will cause the disease hemochromatosis. Okay. Now, we were talking about this ferrous turning to ferric form with the help of hepcidin. So this ferric form also is not allowed or is not able to move freely in the blood. It needs a transporter. So there will be a transporter which is called transferrin, thankfully. Transferrin, which will help in the transport of the ferric iron within the blood. So this is the first raw material which is required for the process of erythropoiesis. So we have discussed about which type of ions are we are getting through the diet, and which are the normal oxidation states which the iron exists in. So we can do a quick run through this. Okay, so normally in our diet there are two type of ions. Which are they? It is heme iron and non-heme iron. The heme iron, which we are getting from the meat based diet, and the non-heme iron, which is getting from the vegetable based diet. And the heme iron is Fe2 plus or the ferrous form, and the non-heme iron is Fe3 plus or the ferric form. So the Fe2 plus iron can be easily absorbed or easily led through the cell membrane with the help of a divalent metal transport 1 protein. But for the Fe3 plus or the ferric ion to pass into the cell, it will have to convert into Fe2 plus so that it can pass through the divalent metal transporter 1 protein. So it will with the help of cytochrome reductase B enzyme, the duodenal cytochrome B reductase enzyme or the protein, it will be converted into what will be the Fe3 plus will be converted into Fe2 plus, and which in turns will go into the cell with the help of DMT1 protein. So after getting into the cell, there will be another protein which is apoferritin, to which the Fe2 plus will attach and form ferritin, and this ferritin will cluster together to form hemosiderin. Okay, so then the iron from this pool of iron, which is hemosiderin, okay, it will go out into the bloodstream so that it can get to the bone marrow for the process of erythropoiesis. It will move out through a special protein in the cell membrane, which is called ferroportin. Okay, from this ferroportin the Fe2 plus comes out, but as I said earlier, the Fe2 plus is not allowed to accumulate within or outside the cell because it can cause free radical reactions to happen. So this Fe2 plus should be converted into Fe3 plus, which is done by another protein on the membrane which is called hepcidin. So out of this hepcidin and ferroportin, the ferroportin is kept under check by a protein called hepcidin, which is produced by the liver and is controlled by the HFE protein. Any mutation of this HFE protein or the lack of production of HFE protein will cause the reduction or the lack of production of hepcidin, which in turn will cause the over accumulation of Fe2 plus and in turn Fe3 plus in the blood, which is a condition known as hemochromatosis. Okay, so without any problem, if the hepcidin is present and the ferroportin is working normally, what happens is we'll have enough amount of Fe3 plus in our system. So this Fe3 plus should be transported with the transport protein, which is transferrin, which will help the transport of Fe3 plus inside the blood to the bone marrow or whichever site we want it to reach. So that is about the iron metabolism and intake, and it is the first step of erythropoiesis. Hope you guys understood something from this. I know this is not the best video out there, but I promise you guys that I'll definitely work on these videos and I'll make it better to understand, and also I'll make it much more engaging than this. I just needed to start somewhere. That's the importance of this video. And if it's able to help you guys with some concepts or some theory, anything, I'm happy. That's really an added benefit for me. So next time I'll try to improve. So until then, bye bye. See you guys.